Good morning, students. Today we can start our class with the next part of this chapter, that is nerve impulse generation and propagation. In the last class, we have learned about the structure of nerves, what are neurons, what are glial cells, everything we have learned. So we can go to this topic today. First thing we need to understand to learn about the impulse generation and propagation is a stimulus. What is stimulus? Stimulus is what? It's a force that excites the neuron. A force capable to excite a neuron, that is stimulus. It may be of electrical, chemical, or mechanical. That stimulus can be electrical, chemical, or mechanical. Any type of force that can excite a neuron is called stimulus. Then coming to resting membrane potential. That means the potential of the membrane, any membrane, when it is in resting condition, when it is not stimulated, that is the resting membrane potential. So, what is resting membrane potential? It is the potential difference across the neuron membrane, across the neuron membrane wall. Listen here, when considering the charge of the cell membrane, always the outside side of the cell wall is positive. For not get it confused with whether it is negative or positive, one trick is here. That is, we need to remember the code OP. OP means outpatient in hospitals. That code we can rethink as out OP. O for out, outside, P for positive. So every time outside is positive. It's a small trick to not get confused over whether the outside is positive or negative. Okay. So every time outside of the cell wall is positive and inside is negative. So there is a potential difference. That potential difference across the neuron is called resting membrane potential, and it is measured using microelectrodes. It is measured using microelectrodes. Theoretically, the value of resting membrane potential is minus 90 millivolt. It is only theoretical, it's not actual. Yeah, this value is obtained by assessing the ionic concentration across the membrane. Experimentally, the obtained value is minus 75 millivolt, much higher value than this minus 90 millivolt. Okay, this is the experimental value, and we are always speaking about value around this one. Minus 75 millivolt is the resting membrane potential. Here's the picture to show you the charge distribution, the outside and the inside region of membrane wall. Okay, we can go to next slide. Factors contributing to the resting membrane potential. Which are the factors that helping to maintain this minus 75 millivolt always constant whenever the membrane is not excited? Which are the factors helping? First one, presence of certain fixed anions, presence of ion leak channels, and also because of presence of sodium potassium pump that we already learned in which chapter? I have discussed with you the functioning of sodium potassium pump. I think it is in digestion, right? Right? Okay. Then we can go to fixed anions. What is fixed anions? Fixed anions are certain positively charged substances. Which are the substances coming under that category? They may be of proteins, carbohydrates, nucleic acids. These substances are all abundant inside the cell and their charge is what? Negative. They are anions, and because of their bigger size, they cannot diffuse out of the cell. So, they always remains inside the cell and their charge is negative. So, they are called fixed anions. So, negative charge carrying more substances are present inside the cell that imparts a negative chargeability towards the inside of the cell wall. Right? That factor supporting the maintenance of negative charge inside the cell. Then coming to the ion leak channels, how it helps. Ion leak channels are some channels, some openings found in the cell membrane. And they are of sodium and potassium leak channels. Both sodium and potassium ion leak channels are present in the membrane. This is distributed. And the number of potassium leak channels are more when compared with the number of sodium, which is more potassium is not here. Potassium ion leak channel number is more when compared with the sodium ion leak channels number. And as we know, which ion is abundant outside and which is abundant inside of the cell? You know, because I already told you in some other chapter. I think it is in digestion and nutrition chapter itself. Which ion is more inside and more outside? Sodium or potassium? Potassium is more inside and sodium is more outside. So what is the normal trend? Normal trend of flow from a region of higher concentration to a region of lower concentration. See? So, what way the ions flow? Now, potassium always try to flow from inside of the cell to outside because inside potassium's amount is more, higher concentration. So, always potassium tries to flow from inside of the cell to outside the cell. Then what about sodium? Sodium is more outside. So, its trend is to move from outside of the cell to inside of the cell according to the concentration gradient. For that flow is facilitated by ion leak channels with the presence some carriers, presence of some carrier proteins. That channels are called ion leak channels, and the number of sodium leak channels is less than that of number of the potassium leak channels. That means what? That means more potassium is moving out than the number of sodium that is moving into the cell. Right? Understand? In another way we can think about that. That is the ions that moving out of the cell membrane. Which ion? Potassium ion. What is its charge? Positive charge. More positive charge is moving out. And which ion is moving in from outside? Sodium. Its charge also positive. Less positive charge is moving into the cell. More positive charge moving out and less positive charge is moving in. Why? Because the number of potassium leak channels is more when compared to the number of sodium leak channels. Then again, that factor, that action supports their maintenance of positive charge on outside and negative charge in the inside. Why negative charge inside? Because more positively charged potassium is moving out. The number of potassium ions moving out is not equal to the number of positively charged sodium moving in. So, that way negative charge inside can be maintained. I think you understand. Then coming to sodium potassium pump. Sodium potassium pump always work against the concentration gradient and it always attempt to export the sodium ions that comes towards the cell to the outside, right? Think it. What sodium potassium pump actually doing? It is pumping out sodium and pumping in potassium against the concentration gradient, right? And what is the number? What is the amount they are importing and exporting? They are exporting, they are moving out 3 sodium ions at one time. At the same time, they are importing only 2 potassium ions. Right. If all are by heart about the sodium potassium pump function, then clearly understand what I said to you. What is the quantity wise, number wise, what is the difference? 3 sodium is moving out and 2 potassium is moving in. Against the concentration gradient. Again, what is happening here according to the charge wise? More positive charge is moving out and less positive charge is coming in. Again, supporting the maintenance of negative charge inside the cell wall. In all that way, that resting membrane potential is maintained with the help of these factors, fixed anions, ion leak channels and sodium potassium pump. Then we are coming to all about this. I have already discussed with you is a picture of ion leak channel. Allow constant flow of ions by facilitated diffusion. Both K plus and sodium leak channels are present. K plus leak channels numbers are more. In significance, I already told. Significance what? Positive charge is more out and negative charge can be more inside the cell. Next slide. This is sodium potassium pump. Sodium potassium pump is always doing 3 sodium is moving out and always 2 potassium. Again here more positive charge is transporting towards out and only very less positive is moving in. That's why it supports the existence of negative charge, negativity inside and positivity outside. In action potential. It is a transient change in resting potential. Transient means what? Not a permanent change. It's a temporary change to initiate some response, to stimulate a nerve. For stimulating the nerve, the action potential should be generated in the nerve, one point of the nerve. And that action potential need to be conveyed or transmitted down the nerve for transmitting that signal. Maybe up to the central nervous system or from central nervous system to the effect. Anyway, for conveying the message, action potential should be generated. That action potential is generated with the help of a stimulus. And we are going to see how the action potential is generating, what the process. So membrane, whenever a threshold stimulus is occurred, threshold means enough strength stimulus. A stimulus having enough strength to stimulate a nerve. That type of stimulus is called a threshold stimulus. Okay, whenever a threshold stimulus is gained by a nerve, what will happen? First, voltage gated sodium channels are opened. Voltage gated stimulus, whenever that stimulus came, its presence is sensed by voltage gated sodium channels and it is open. When it is open, what will happen? Sodium is flushed towards inside because sodium's amount is more outside. High concentration here and low concentration here. So whenever a hole it found, it rushed towards inside. Okay. Then what happened? Depolarization. A charge reversal is happened on that particular point. Right. Here, as against the normal condition, some point of here become negative and some point of here is changed to positive. Here actually positive is required, but it becomes negative, and here actually negative is required. Here it is changed to positive because of this additional flush of sodium. After this flushing, sodium channels closed, and at the same point itself, very near to this point, present some potassium channel. For making you to understand it clearly, I draw these channels apart from these channels. Actually, it is very close to this and the same point of this depolarized region. For your understanding, better understanding, I draw these channels some distance apart from here. Okay. At the point when depolarization happened and sodium channel closed, at the very moment itself, potassium channels open. If potassium channel open, what will happen? Potassium's concentration inside is high and outside is low. So, potassium will move out. That will result in repolarization. Again, maintain that all the resting membrane potential. Again, the charge will be converted to the normal state. Now, what is the whatever the normal state, that into that state the charge is again maintained, again created because of the potassium channels opening. Okay, then a depolarization and repolarization happen. And at the for a short second, at this point when repolarization happen, a peak hyperpolarization occurs because of the overshooting of potassium. More potassium is moved out. So a high depolarization is observed at the very next moment, first few second moment of repolarization. After that, it will be comes to our normal resting membrane potential, that is minus 75 millivolt. So that is the generation of an action potential. Here, a depolarization and a repolarization happen. And depolarized condition is an action potential. And again it comes to a repolarized state. That is that change, the cycle of change happened in a particular region of neuron. And these changes are repeating to next region, next region, next region. And in this way that stimulus is conveying to the entire length of the neuron. I think you understand that. So, action potential, how it is developing with the help of stimulus. When stimulus came, what will happen? Voltage gated, sodium channel open, sodium flush. That as a result of this, depolarization happen. After depolarizing, sodium channels are closed, and with the closing at the same moment, at the same spot, potassium channels are opened, and it results in the flushing out of potassium ions, and which in turn results in the repolarization of membrane again to the resting membrane potential. And in the next point again, which one open? Sodium, voltage gated sodium channel open, sodium flush towards inside, depolarization happens, and after a certain level is attained, sodium channel closed and the potassium channel is open, potassium is flushed out, repolarization happens. Then again to the next point, voltage gated sodium channel again open. In the same way, the process is conveying, repeating towards the entire length of the neuron. This is the process of nerve impulse transmission. Okay. Then we are going to next term. Threshold stimulus and potential. What is threshold stimulus? I already told you what is this. What is the minimum strength required for a stimulus to elicit an impulse? That enoughly strength stimulus is called threshold stimulus. Threshold stimulus is such a type of stimulus having enough strength to elicit an impulse. Enough means minimum. At least that minimum strength should be there to elicit an impulse. The normal action potential's value is minus 65 millivolt. Next is latent period. Latent period means it is the interval period, the period between application of the stimulus and the initiation of an action potential. Some small, minor, very minute time interval is existing between the application of stimulus and the initiation of action potential. That period is called the latent period. Then all or none law. All or none law means then when only enough strength is there for stimulus, then only there is a generation of action potential. Otherwise, there is no action potential. There is no change to the nerve. All or none law. Whenever enough strength is there. If the stimulus is enough strength, then there is generation of action potential. Otherwise, no action potential. That explains in this law. All or none law. If it came, it come in all. Otherwise, none should be there. All or none law. Then propagation. How is it propagate? By the cyclical changes of depolarization and repolarization. Depolarization and repolarization. That cyclical change convey the action propagation. Propagation of action potential towards the end of the nervous system. Then what is all these points, all or none law, latent period, threshold stimulus, refractory period, all are very, very important questions, 2-mark question, 1-mark question for the university examination many times from this chapter. So, consider these points like that. Then refractory period. Refractory period means period in which a nerve will not respond to a stimulus after eliciting an action potential. That means, consider the case of us. If we do a great effort work and we are exhausted and we are very tired, and during that time we can't do any further work. That period, that tired period, it's called the refractory period. When coming to the condition of the nerve, if a nerve, the condition, the period of that nerve after an excitement, after an action potential, that period, that nerve cannot be elicited by a second impulse. That inactive period is called refractory period. Understand? What is refractory period? Period in which a nerve will not respond to a stimulus after eliciting an action potential. And two stages are there. Absolute refractory period and a relative refractory. Absolute refractory period means during this period, it is in no way it is not possible to elicit a second action potential. Then relative refractory period is a period in which sodium channels are gradually reactivating. Sodium channels are gradually reactivating. Possible to elicit another action potential. Some possibility is there to elicit an action potential when nerve is in the relative refractory period. If it is in the absolute refractory period, there is no chance for eliciting a second impulse. Next, we need to learn about saltatory conduction that we can take in the next section. Okay, with this we can finish today's section. Thank you.